Okay, after looking at the structures of the bacteriophages, right, now we're looking at the reproductive cycles of the bacteriophages. So in the previous video, we already mentioned, right, the T4 phage reproduce only by the Lytic cycle. So now we look at how the Lytic cycle is carried out. Okay, so the first step is to attach the virus to the host cells. Right, so the attachment is through the tail fibers and the base plate of the virus, which will recognize and bind to receptors found on the surface of the host bacterial cell. Right, so this recognition and binding, of course, is through complementary shape and charge, right, between the proteins, right, of the tail fibers and base plate, with the specific receptor proteins on the host bacterial cells. Okay, after that, the next step is the entry of the viral genome, right? So it's not the entry of the virus. The virus doesn't really enter the host cell for bacteriophage. Only the viral DNA enters, and that is through the contraction of the contractile sheath, which will then push the hollow tube that is found inside the tail. You push out the hollow tube, and the hollow tube will puncture, right? Puncture through the cell wall and cell surface membrane of the host bacterial cell. So the hollow tube will enter, right, puncture inside the bacterium, which will then allow the viral DNA, right, to enter the bacterium through this hollow tube. Right, so that is the start of what we call the eclipse period, which is the start of viral infection. Okay, so when we talk about entry of viruses, it's really not talking about entry of the virus body itself, right? It's actually talking about entry of the viral genome, right? Same for the other viruses, right? The point of entry, the important part of entry is actually how to get the viral genome into the host cell. Okay, so in this case, the virus body, the capsid, the tail, right, doesn't really enter the host cell. Only the viral DNA enters. Okay, so this is a electron micrograph showing you this process. Right, look like some people with big heads squatting down doing business, but not really. So this is the bacteriophage, right? This is the hollow tube, right, and the DNA enters the host cell through the hollow tube. Right, so from the capsid, right, we'll go through the tail, go through the hollow tube, right, into the host cell. Okay, then once the viral DNA is inside the host bacteriums, then the viral DNA will be transcribed and translated. Then that will make your viral proteins, right? And of course, the viral DNA is also replicated, right, to form more copies of the viral DNA. Yeah, so two functions. The viral DNA serves as template for DNA replication, also serves as template for transcription and subsequently translation. Okay, one of the viral protein or enzymes that is produced through the protein synthesis using the viral DNA is actually a enzyme that will hydrolyze the DNA of the bacterial cell. Right, so the bacterium, right, or the bacterial cell cannot then continue its own protein synthesis, and all the raw materials like amino acids, all the energy in the form of ATP, all the ribosomes, all the RNA polymerase will now be used to transcribe and translate the viral DNA to make viral proteins. Right, so basically the virus is taking over the protein synthesis machinery of the host cell by degrading the host bacterial DNA with one of the viral enzyme that is synthesized. Okay, so once the rest of the viral components, right, the viral DNA, the capsomeres that forms the capsid head, all the other proteins that form the tail, right, once all these are synthesized, then they will be assembled into mature bacteriophages inside the host bacterial cell. Right, so you can see this in this diagram, right? All the viruses are assembled inside. Yeah. Then the last step, of course, is for the newly assembled viruses to leave the host cell. Right, so that is done through another viral enzyme that will be produced. It is a phage-coded lysozyme or a viral lysozyme. A lysozyme is an enzyme that breaks down peptidoglycan, right? So that will then digest the peptidoglycan cell wall of the host bacterial cell. Once the cell wall is damaged or hydrolyzed, right, then water which usually enters the cell, right, because the inside of the cell is more concentrated, have a lower water potential. So usually the bacterial cell doesn't burst because of the cell wall, right, protecting itself. But now the cell wall is damaged or hydrolyzed, so then the bacterial cell will burst or lyse, right? We call this lysis or burst. So once that happens, right, then the bacterial cell breaks apart, right? So that's where the newly assembled viruses can leave. Right? So that's the exit or release of the viruses, and that would be considered the burst period. Right? So we have eclipse period, we have burst period. We'll look at it in the graph later. Okay. So this is the graph. So the eclipse period is when the virus first attached to the correct. First attached to the host cell, and the viral DNA enters, right, the host cell before the phages, right, before the new viruses are made inside the cell. Yeah. So that's where the attachment, the entry of the genome, right, the viral DNA, also the replication, right, making of new viral DNA, making new viral proteins. All this happen during the eclipse period before the new viruses are assembled inside the host cell. Okay. So attachment entry, right? This is where the first assembled virus appears inside the host cell. So you can see this one. This is the point where the extracellular phage increase, right? So that would correspond to the release, right, of the phages. So this is where the exit step happens. Right? So that means in between here, right, is the further assembly of more new phages inside the host cell. Right, so this will be the end of release when the number outside the host cell does not increase anymore. Okay. Right, so in terms of the processes that happen, right? So these are the processes that happen within each of the periods in this graph, which I have mentioned just now already. Okay. Right, so that was the bacterial phage, T4 phage, right, with the Lytic cycle. Now we have lambda phage with both the Lytic and Lysogenic. So not just Lysogenic, but both Lytic and Lysogenic cycles. Okay. So the Lysogenic cycle in particular is a phase of the reproductive cycle that replicates only the viral DNA without killing the host cell. Because the Lytic cycle, you see, at the end of the Lytic cycle, the host bacterial cell will burst, right? And then of course the cell die. Right, so the Lysogenic cycle doesn't do that. Right? So the bacterium or the infected bacterial cell is happily living, but with the viral DNA inside, and the viral DNA will be replicated inside the host cell. Okay, so in terms of the process, the attachment is the same, just that lambda phage, of course, does not have so many tail fibers, right? So the binding is a bit different, but still there will be binding, attachment to specific receptors. There is still the contractile sheath contracting, driving the hollow tube inside the bacterial cell. The DNA enters through the hollow tube. So these two steps are the same, right? One difference is once the viral DNA enters the cell, it will circularize. So this one is quite specific to the lambda phage, which does not happen for the T4. After that is the choices is a branch off of two different reproductive cycles, right? So the Lytic cycle can continue right after the viral DNA has entered, right? So if it continues with the Lytic cycle, then it will be just similar to the T4, and there will just be Lytic cycle with no Lysogenic, right? So just Lytic cycle, yeah. However, there is also the choice, right, or the possibility that the Lysogenic cycle is adopted after the entry of the viral DNA. So in this Lysogenic cycle, right, the viral DNA gets integrated into the bacterial DNA or bacterial chromosome, right? So as you can see here in red, if you can see the color, right? So the viral DNA will be inserted in a site, right, in the bacterial chromosome. Okay, in this case, then the naming change. We will call the integrated viral DNA a prophage, and this bacterium carrying this prophage is known as a lysogen. So when you see the word prophage, then you know this is referring to the viral DNA that's already integrated into the chromosome of the infected bacterial cell. Okay, most of the genes in the viral DNA are not expressed or not transcribed. Only one is transcribed and expressed. That is the gene coding for a repressor protein. So this repressor protein will, the gene coding for the repressor protein will be transcribed. The repressor protein will be translated by the ribosomes of the host bacterial cell. And this repressor protein will then function to stop the expression or transcription of the other viral genes. Okay, so really only one of the viral gene in the viral DNA is expressed at this stage during the lysogenic cycle. Okay, so then right now you can see why the bacterial host cell is not killed, right? Because there's no new viruses being made inside the bacterial cell. Yeah, so it's only the viral genome, viral DNA, that's integrated into the bacterial DNA. So the bacterium will happily live and continue to divide, and when the bacterial cell divides, the bacterial cell will replicate its own DNA, right? And then the viral DNA will be replicated together with the host DNA of the bacterial cell. So when this one parental cell divides into two daughter cells, right, both daughter cells also contain the viral DNA or the prophage. Okay, so just with one virus, right, infecting one cell, after the cell divide, now there are two bacterial cells containing the prophage. So as you can imagine, right, when these two cells continue to divide, then there will be more and more cells that contains the prophage. So that's the beauty or the advantage of the lysogenic cycle. Okay, and the lysogenic cycle can be induced to return to the lytic cycle, usually by extracellular stimuli, which are usually detrimental to the bacteria. So the purpose is this, right? In the presence of all these detrimental substances or conditions that will kill the bacterium, before the bacterial cell die, right, the prophage or the viruses want to be made, or you want to make the viruses. If not, once the bacterial cell die, right, the prophage, the viral DNA, will be gone together with the dead bacterial cell. Yeah, so before the bacterial cell dies, the virus want to reproduce and create more viruses that will leave the bacterial cell before the bacterial cell die. And so that's why usually the trigger to return to lytic cycle are certain stimuli that are detrimental to the bacterial cell. Okay, so when the detrimental conditions arise, right, what happens is the bacterial cell will produce proteases as part of the bacterial cell's own response, right? And the protease will then hydrolyze some proteins, right, inside the bacterial cell. One of the proteins being hydrolyzed is actually the repressor protein that that is coded for by the viral DNA. Yeah, so once this repressor protein is hydrolyzed, right, then the transcription and expression of all the other viral genes are not stopped. So that's where then the viral gene that codes for capsomeres will be transcribed, and capsomeres will be made, right? All the other viral proteins will be made. The lysozyme, right, the viral lysozyme will also be made, right? So that's where then the lytic cycle happens, right? So the other viral components will be made, right? Viral DNA will be replicated, then the new viruses will be assembled, and again there's a lysozyme, right? So the virus will burst because of the degradation of the peptidocyte cell wall, and then the new lambda viruses will be released. Right, so that's switching back to the normal lytic cycle. Okay, so this is how the lambda phages reproduce by both the lytic cycle and the lysogenic cycle.